Hello and welcome back to the Metabolism series. Today's video is going to be a little different. We're going to view how the signaling and the transport through the membrane and through the cells occurs. So, without further wait, let's begin. Okay, so we are here and we have two topics today. We have transport and signaling. Transport. Okay, transport through the cell membrane. We have to define what the plasma membrane does. Its functionality is that it separates the living from the dead and keeps the living alive. Okay, so the role of the plasma membrane is to keep those organelles inside the cytoplasm and define the cell itself. In transport, we have many classifications, and if we start, we have the classification by direction. When a molecule enters the cell, a single molecule, it is called uniporter. If two different molecules enter at the same time, it's called symporter. And when one enters, the other exits, it's called antiporter. Then we have the classification by charge, voltage dependent or not, because some channels or transporters need voltage to function properly and others simply don't. They use other mechanisms such as ligand, for example. Then by action, it can be passive or active transportation. Passive transportation occurs with a gradient concentration, and active transportation requires ATP energy and goes against that gradient. If we classify transport by mechanism, we have channels and protein transporters. The main difference here is that protein transporters can go in both ways. Channels, on the other hand, can only go one way. Okay, so transport types. Simple diffusion. Simple diffusion is done by small polar uncharged molecules that can pass through the cell membrane without any enzyme following a gradient concentration. They go from where is high concentration to low concentration because they try to equalize those concentrations. They have the purpose to create balance between those differences. So then we have channels. Channels make a selection of the ions by size. For example, in the sodium channel, that only sodium can enter because it's so small that only sodium can go through. Or by solidation layer. And here I put potassium. Why potassium? Because potassium is bigger than sodium. But if the potassium channel was with selection by size, sodium, as being smaller, can go through this channel, and we don't want that. So, as ions travel hydrated, the dehydration of potassium in his channel is what it allows him to travel through and block sodium, as the solvation layer is much bigger in this ion, in the potassium, and the dehydration can occur only on the potassium. That's a very tricky solution the cells find out. And then we have opening signals of channels, such as voltage, we already discussed that, ligands, light, temperature, and pressure. All these things, if they change, they can open a channel. And then we have transporters. Transporters dependent on specific binding sites. What does that mean? That means that they act like an enzyme. They have a specific site where a molecule, a ligand, gets bind to, and transporters allow both directions. As in ATPases, when we have ATP, we can get ions against the gradient, and when we don't have ATP and ions go through the gradient in the normal way, ATP is created. And some examples of transporters are the sodium-potassium ATPase, this is very popular, the sodium-glucose transporter, the calcium pump, the sodium-calcium transporter, the ABC transporter, lactose permease, and GLUT. GLUT is glucose transporters. If you remember from the video of glycolysis, we said that GLUT2 is in charge of getting glucose inside the liver, and GLUT4 is insulin dependent and gets glucose inside the muscles, for example. Okay, so now let's go with signaling. Signals trigger responses. Responses such as proliferation, apoptosis, which means cell death, metabolic change, and mechanical response. Signaling is between cells, and transport is on the membrane of the same cell. So we have here in the types of signals, they can be neurotransmitters and peptide hormones that can go through the nervous system, steroid hormones, thyroid hormones, retinoic acid, and vitamin D. Those are very common type of signals we have in our body. Then we have the types of receptors, because a cell sends a signal to another cell, but the receptors are needed to get that signal and to be able to interpret it well. So we have internal receptors, which are cytoplasmic and nuclear. That means that they can be inside the cytoplasm or inside the nucleus of the cell. They regulate metabolism and gene expression. The binding site, DNA binding domain, regulatory domains. What does that mean? They have a binding site for the molecule to bind to. The DNA binding domain, that means that the nuclear receptor then acts on the DNA and regulates the gene expression. And then we have the regulatory domains. Those are domains of the same DNA that are regulating the gene expression and also the signaling. On the other hand, we have external receptors, which are ion channels, we discussed them earlier, catalytic membrane receptors, that simply they have an enzyme on the membrane and they speed up a reaction. The G protein coupled receptors, these are very interesting, they are very important as well. What they can do is activating channels. They can synthesize cyclic AMP and they can send the signal to get shut down. The cyclic AMP works as a second messenger and amplifies the signal. A second messenger is what amplifies the signal inside the cell. So for example, it gets bind to the G protein, which is on the membrane, and then via the cyclic AMP the signal gets amplified to all the rest of the cell in the cytoplasm. Other second messengers are inositol phosphate and deacylglycerol, and also arrestin. Arrestin is very interesting because it does the contrary thing, which is signal shutdown. Signal amplification over-exaggeration, when there is too much second messenger that amplifies too much the signal, we reach an adrenaline cascade, for example, or epinephrine cascade, and kinase cascade. Those cascades are simply an over-exaggeration of a normal signal. They can lead to many things, we will see them later. The tyrosine kinase receptors. These type of receptors have the possibility to phosphorylate themselves. Tyrosine kinase receptors are linked to kinase cascades, as we saw earlier. Those are the same cascades, and we need them to regulate various processes in the body, such as when, for example, we have insulin present in the bloodstream and we need to quickly create insulin receptors, because if not, we are not going to be able to metabolize glucose properly. So there are some cases when our body needs something very quickly, and we use the cascades, kinase cascades, but as well adrenaline cascades, to quickly get those things done. That's a very effective way and very fast way of signaling and to making reactions to occur fast. At the same time, we need something that needs to shut down those cascades, because here's the thing: when you create a kinase cascade, for example, it's a cascade. It's not going to stop itself, especially in tyrosine kinase receptors, because they have the capacity of phosphorylate themselves. They're not going to stop. And imagine how bad can that be, because if we have insulin present and we start creating insulin receptors and everything is fine, but then we don't need them anymore and we keep creating them, that's not good. So we need to stop them, and there are some mechanisms of signal shutdown. For example, with the arrestin. And this consists in removing phosphates from kinases, as these ones, and or removing membrane receptors once activated. So we say, okay, these receptors of insulin in the membrane have been activated, they are no longer needed, and we are going to deactivate them. And that's how signaling works in our body. That's it for this video. I hope you enjoyed, you understood it well, and I will leave the image down below in the description so you can download it. It's yours, study it very well, and that's it.